with a beta hairpin loop, which is just a structure of the protein, but it forms a binding pocket in which ATP is able to bind, which is important because when calmodulin binds to this site, it leads to capsin, which is an activator of this protein, to be desensitized. So when ATP is competitively binding over the calmodulin, it allows the capsins to still activate the TRPV. So in this way, the acrine repeat is important because it keeps the sensitivity of the TRP up instead of when calmodulin is able to bind to the same site, it leads to the desensitization of the protein. So it's important for keeping the protein at main function and not getting desensitized, which TRPM doesn't have. So that'll kind of be shown later, which in the diseases, they can be desensitized. So going into how it's activated by temperature directly and indirectly. So it's able to activate by temperature directly because the temperature shifts the voltage-gated dependence of the channel directly. So kind of like with calcium channels and stuff like that, if there's a positive on the outside of the cell, it'll cause the channel to open. So this is kind of the same thing, but instead of a positive or negative, it's the temperature that's opening the channel directly. And then indirectly is a secondary messenger. So the temperature leads to a secondary messenger inside the cell, and then that will in turn change the cell. And then one of the mechanisms that it does this by is redox signaling. So this happens also directly and indirectly. So it's able to happen directly by changing an amino acid modification on the TRP, and it's an oxidative amino acid modification. And again, it's indirectly through a secondary messenger where it'll show here with the direct amino acid modification, it uses hydrogen peroxide to lead to the oxidation. So those are just a couple different TRPs that were shown to use this direct amino acid modification. And there's a study done where they used PAGE, electrophysiology, mass spec, just a couple different techniques. And they were able to find a disulfide bond between these two cysteine residues. Specifically, it was cysteine 258 and 742. And then when they treated this with hydrogen peroxide, they found that the cysteine 258 was oxidized. So then that led to the break of the disulfide bond between those two residues, and that led to thermal hypersensitivity of the protein. So where this was in TRPV1, where it's typically sensitive at 43 and above degrees Celsius, it moved it to a lower temperature so that the calcium dependence was opened at a lower temperature. And then an indirect secondary messenger, adenosine diphosphate ribose and cyclic adenosine diphosphate ribose are made through hydrogen production. So when hydrogen peroxide is added to the cell, then these things are made, and these can be seen as a secondary messenger. And these were acting on TRPM2. And this is interesting because cyclic ADPR is previously known as an insulin secretion regulator. So it's interesting that when this acts on this cell, the heat-evoked activation of this is connected with the cell's ability to increase insulin secretion. Although I had no idea about this before, I just thought that it was really interesting that those two things were able to come together, and this kind of shows how homeostasis is conserved in that way. And then talking specifically about calcium homeostasis. So why this is important is because the influx of calcium caused by these receptors leads to homeostasis. So this happens by activating protein kinases, which in turn change the gene expression of the cell, and then also just the TRP's interaction with other calcium sensing protein also helps maintain calcium homeostasis. So these were the different TRPs that were seen specifically with activating protein kinases. And this is just all, like, TRPs in general are able to associate with the endoplasmic reticulum, and that in its interaction with the mitochondria leads to homeostasis. So looking at the protein kinases, there was a study done where they activated TRPM3 with this progyline sulfate, and they saw that this activated MAP kinases. The MAP kinases activated these ERK, which are extracellularly regulated kinases, and then this happened to activate the synthesis of EGR1. And this was interesting because EGR1 is correlated with neuronal cell death, reproduction, proliferation, and it just shows that this is how this TRPM3 is then able to change the fate of the cell. So if there's excessive calcium release and there's a lot of this EGR1 produced, then it leads to the control of neuronal cell death where that isn't there if this doesn't happen. And so then this is just a schematic kind of to show you guys a little bit more about how this happens. So the progeline sulfate activates this TRPM3, which then lets the calcium come into the cell, and it can bind specifically to PKC, which activates the ERK, and then this leads to activation on the EGR1 gene, which then it can be transcribed and translated and then be a working protein in the cell. Another thing it does is its correlation with the endoplasmic reticulum and the mitochondria. So the calcium release of the endoplasmic reticulum specifically depends on 3 different things. So there's this specific SERCA, so it's the calcium uptake of the sarcoendoplasmic reticulum specifically that changes the calcium release, but then also binding proteins on the lumen of the ER, but also the activity of calcium release channels on the membrane of the entire cell changes the calcium release of the endoplasmic reticulum. And that's where the TRPs come in. So under normal calcium release from the ER, the mitochondria goes through the citric acid cycle and makes energy and your cell lives. But if there's excessive calcium release from the ER, then cytochrome C is released, and this causes apoptosome and capsaicin activity, which leads to death. So again, here's a little schematic that kind of shows how this happens. So there is an inositol triphosphate receptor right here that specifically links the ER to the mitochondria. So that shows the association. If the ER is producing low calcium levels, then the mitochondria goes through citric acid cycle, also known as the TCA cycle, and it makes ATP, and then this leads to the survival of the cell. But it causes death if there's very high calcium levels, because then the calcium is put into the mitochondria, and it causes increased reactive oxygen species in the mitochondria, which pushes down the TCA cycle. And then it leads to the cytochrome C, apoptosome, and capsaicin activity, which then leads to the death of the cell. So back to the diseases, now that we have a little bit more background on what TRPs are and what they do. So these mutations are linked to human diseases. So as talked about before, the angiotensin VSMC malfunction. So TRPM8, the cold-sensitive TRP, there's a downregulation of this protein, which leads to increased ROS and hydrogen peroxide production. And prostate cancer is kind of the opposite, it's TRPM8 upregulation. So the angiotensin-induced vascular smooth muscle cell malfunction, there's a study done where they took rats and they gave them a 2-kidney, 1-clip operation, which is basically just a hypertension model for the angiotensin. And they found that there's a 60% reduced mRNA and protein of the TRPM8. But when they added menthol, which is an agonist of this receptor, they found that the TRPM8 was activated, and then this reduced the ROS levels in the cell. So it reduced the hypertension that was then found in the cell. Now, prostate cancer, just in the past, they used cancer patients and they found an increased sensitivity to menthol. So then this suggested that there is a TRPM8 overexpression. And this has just been used in the past for more of a diagnostic and prognosis of prostate cancer. But I propose that it could also be used for a search of treatment for prostate cancer. So Voakin gene is a chemical that is found in a root bark of a tree in Africa. And it's been used in the past just for treatment of diarrhea, convulsions, edema, and madness. So a couple of different things, that I thought this was a very interesting compound because it's known to inhibit tumor-induced angiogenesis, which is important because angiogenesis is the formation of new blood vessels. And when a tumor is trying to grow, it needs these blood vessels to get nutrients to it so it can keep growing. So this compound is able to inhibit this, so then the amount of tumor cells goes down. But there's also a study done where they found that this Voakin gene also competitively inhibits the binding of menthol to TRPM8. So I thought this was interesting to maybe possibly use this as a treatment for prostate cancer because it's not only known to inhibit the angiogenesis, but it also could be seen to inhibit this TRPM8 function. So just a quick recap, so TRPM is the cold sensitive, and that is what is typically seen with our diseases. TRPV is hot sensitive, and that has the anchorin repeats, so it's very regulated and don't see as many diseases with it. It's able to be activated directly and indirectly by temperature, and indirectly by hydrogen peroxide. So it's obviously important that both of these be able to be activated directly, because if you do, say, climb a mountain and you put your body into very extreme temperatures, you want a quick response to be able to maintain homeostasis. And it's able to maintain that homeostasis by the protein kinases in association with the ER. And as I was just saying, the two diseases that it can be seen with, TRPM8 downregulation angiotensin, which meant that was an activator, and then prostate cancer, where TRPM8 is upregulated and possibly voacagine is an inhibitor. So my proposal is possibility of using voacagine as a treatment for prostate cancer. I propose that in an experiment done with this, that we would monitor massive prostate cancer cells after the addition of a placebo, voacagine, this voacagine combined with other known prostate cancer treatments, and then just other known prostate cancer treatments. And I want to use this by monitoring the cell with quantitative mass spectrometry. So this quantitative mass spectrometry is important because it can not only identify, but it quantifies the number of proteins and also the modifications from biological material. So just in the past, prostate cancer cells are known to have alterations in this TMPRSS2 ERG translocations and also mutations in all these different proteins. So it's important that we're able to quantify and identify these proteins to see if the cancer cell has changed. And then it's also important to use this because we know that voacagine affects cancer in other ways than just the TRPM8 function. So not only can you see is the cancer cell changing, but specifically is it changing the TRPM8 regulation in the cell. So this is just what a mass spec does. You inject your sample, it heats it to vaporize it, and then it gets ionized, it passes through a magnet, and then it gets diffracted. It gets separated based on its masses. So this is something that you might get out of this. So it's going to have the mass and ion ratio over here, and then the relative abundance here. So if you were to take a mass spec of this right away, you would see the P10 and PSOS and all of those levels very high. And then if those levels had decreased, then you'd be able to see that the percentage of cancer cells in the mass has decreased. But obviously, TRPM8 function is going to be really high, so that's also be something that we would want the relative abundance to go down on. So I propose that we compare the percentage of these cancer cells after about a 2-week treatment. And then if voacagine itself does significantly better than anything else, then awesome. Put it into animal trials. It's a great prostate cancer treatment. But if this combined with other known prostate cancer treatment just does better than other cancer treatments, then it could be used in combination, which is obviously a really important thing because usually you have to do that with cancer anyways. You want to use the radiation to lower the P10 levels, and then you want to use the voacagine to lower the hypoxia-induced factor 1 alpha, which is the angiotensin, and then also reduce the TRPM8 levels. So putting those together would probably be the best idea from what I would guess. And then if the animal trials work successfully, then putting it into human trials, seeing if it works with other cancers, and then yeah, seeing how it goes. But these are my references if there's any questions.
I just had the one.
Yeah.
So you had the disulfide bond oxidation, right?
Yep.
When that occurs, is that channel then, when you revert back to normal conditions, is that protein then effectively inert and cannot be used again?
Nope. Wow. When the disulfide bond is broken, it actually just reduces the temperature needed to activate the channel. But if the disulfide bond is reconnected, then it just goes back to its normal functioning. Yep.
Where is that disulfide bond located in the protein?
It was the cysteine 258 and 752, so I believe it was between the 4th and 5th domain,
Yeah.
so it wasn't directly on.
So it's close to the...
Yes, it's close to the pore, but it wasn't across the pore, yeah.
And for your tumor studies, maybe I missed this, but can you use some kind of cell line and culture to try these? I think you proposed using it in the whole animal model.
Mhm.
Can you use like, I don't know if there's prostate tumor lines, could you do that?
Yeah, so that was like monitoring a massive cell. That was kind of like my first thing that I was planning to use it for. And then if that was successful, then go to the animal trials. But yeah, using the prostate cancer cell line is the first thing to study
Okay,
on.
and then just do an overall mass to see if, I guess I'm not understanding exactly.
Yeah, so, well, I guess I just wanted to have a couple different, like, masses of it, so not just having one cancer cell, but having a couple of them, so you're able to see, like, more of a percentage change, versus then, yes, it's still cancer, and no, it's still cancer. So then so you can better, compare the studies of, the placebo with the Wonkin gene, and then that compared to other things.
Because they have single cell, basically you can do single cell analysis these days. So
Mhm.
like even on a capillary electrophoresis system. So it'd be really interesting to just try single cell analysis if it's possible with a variety of agonists or potential drugs.
Yeah, that could work too. Then I would just obviously want to do like 100 different
Right,
things, just
right,
do
right.
it so many times that you could still get a comparison between the two. But yeah, that's a really good observation.
Yeah, unfortunately, we're gonna have to stop questions there, because we do have to prepare for
Okay,
exam.
thank you.
Because that's